それでは今回も UC サンディエゴとリンク J のコラボレーションに大変ご尽力いただいている UC サンディエゴ国際アウトリーチディレクター、我が宮子様よりご挨拶とモデレーターの紹介をお願いいたします。
こんにちは。Hello, can you hear me?
Yeah, I can hear you.
Okay. So good morning, Japan. Good evening, San Diego. I'm Iwako Waga, Director of International Reach at UC San Diego. And I really am very grateful for Link J and Kyuchi-san and his team for the support of this wonderful webinar series. And I'm happy to report to you that UC San Diego and Osaka University are now forming a new strategic partnership. And we are going to do more collaboration together in research and academia. So just count on us finding great innovations and discoveries together. So now I'd like to introduce Dr. Kinoka Sensei and Andrew McCullough, who will be moderating the session today, and for their brief remarks. And after that, Dr. McCullough will introduce the first speaker. Kinoka Sensei, please.
Thank you very much, Miyako-san. And good morning in Japan and good afternoon in San Diego. It's my pleasure to having providing this seminar. I want to get the many information from the series of this web seminar. So very happy, and at this time we will introduce two big members for the presentation. Before starting the talk, I want to introduce Andrew Sensei from the San Diego. Andrew, how are Andrew Sensei? Hey.
Arigatou gozaimasu, Kinoka Sensei, konnichiwa. It's my pleasure to welcome you to this fourth session of our joint webinar series in Mechano-Driven Functionalization of Engineered Tissues between UC San Diego and Osaka University. My name is Andrew McCullough. I'm director of the Institute for Engineering and Medicine at UC San Diego. And we're delighted to expand our strategic collaborations with Osaka University. Today's session is on biophotonics, and we have two outstanding speakers. It's my pleasure to introduce the first speaker from UC San Diego, Professor Johannes Schoeneberg. Professor Schoeneberg joined UC San Diego just last year after the pandemic started as an assistant professor in the departments of chemistry and biochemistry and pharmacology. So his appointment spans the basic campus and the medical school, the health sciences campus. He earned his PhD at the Free University in Berlin and the Max Planck Institute for Molecular Genetics. And then went to UC Berkeley, where he did two postdocs studying molecular biophysics, microscopy, and theoretical biophysics to build on his computer science training. Now he runs a laboratory on 4D microscopy for cell biology at UC San Diego. And has done an amazing job of getting his lab up and running very quickly. So we're very much looking forward to his talk, which is entitled 4D Cell Biology, Adaptive Optics, Lattice Light Sheet Imaging, and Artificial Intelligence-Powered Big Data Processing of Live Stem Cell-Derived Organoids. Joe?
Hello, can everybody hear me?
Yes, we can hear you.
Perfect, excellent. Share my screen. Thank you, Andrew, for this fantastic introduction. Thank you for having me. I'm really excited to be here today. As I already mentioned, I'm I'm a huge fan of Japan. Essentially, like if you are in optics and if you are in microscopy, then you essentially have to be a huge fan of Japan and Japanese engineering. So you know, almost half of the components that we have on our optical table are from Japan. So yeah, it's fantastic what we're doing today, this lecture series, and starting this collaboration, I think is fantastic. My talk title today, 4D cell biology, adaptive optics, lattice light sheet imaging, and AI-powered big data processing of light stem cell organoids. That's quite a mouthful, but I think what I mainly mean by what is happening here is we are about to see a revolution in the way we do cell biology. And the reason I really believe, and I know that we're going to see this revolution in the way we do cell biology, is mainly because of this. So when you look at the standard way how we come from our questions to our answers in cell biology and also in tissue engineering, then it typically involves two-dimensional cell culture, and it involves some form of two-dimensional microscopy that typically generates in the order of like hundreds of megabytes of data. And then it involves some form of data analysis. You have to essentially get your quantitative analysis from your imaging data to arrive at your answers. And so in all three of these categories, we have these massive technological advances, particularly in the imaging, that are going to bring about this revolution in cell biology. And so let me show what I mean by this. So first, in cell culture, we are now able to grow essentially like tissues, and essentially, which is like the title of this series also. But in my lab, what we do is we grow three-dimensional organoids from stem cells. So we essentially have in the lab human stem cells, and we then differentiate them into small human mini brains. So those are essentially like small pieces of human brain tissue that are models of human neurological diseases. And so, but what is essentially quite sad about this is we never could really image these three-dimensional living tissues live until the advent of lattice light sheet microscopy. And so that is the next big technological advance that happened in Eric Betzig's lab, where the lattice light sheet technology really allows you to do very, very benign and low phototoxicity imaging in 3 dimensions of cells and then tissues. And so then when you actually have this new microscopy technology, and you actually collect 3-dimensional movies of your samples, you no longer deal with like hundreds of megabytes of data, but you suddenly deal with like hundreds of terabytes of data, because like essentially go from like individual planes, you go through 3D volumes that you then collect over time. So we collect massive amounts of data, and at that point you no longer can actually like manually analyze your data, but what you have to do is you have to use big data methods. You have to use high performance computing, and you have to use machine learning to actually quantify your data sets and to get the biological insights out of your data. And so my lab essentially spans all three of those aspects. And I would actually just like to show you like one example of how we can essentially use this new technology and what we can do with this. And so my example is going to be based on something that's called Clathrin-mediated endocytosis. So that is the way where cells essentially take metabolites, hormones, proteins, and receptors from the outside of the cell to the inside of the cell. And that happens by the cell forming these vesicles, essentially forming these Clathrin cages here. They mature, they take these cargo molecules in, and then get cut off from the plasma membrane by these molecular scissors that are called dynamine molecules. And so if you want to study the biophysics of this process of how these vesicles load cargo and then bring it into the cell, what you can do is you can essentially like put fluorescent tags on these endogenous proteins, on like put a red fluorescent protein on Clathrin, put a green fluorescent protein on dynamine. And then what you can do is you can grow these three-dimensional organoids from stem cells. So we put these tags into stem cells, and then we differentiate the stem cells into tissues. And in this case, those were not brain organoids, but we differentiated them into intestinal epithelial organoids. They essentially have this one epithelial layer, have a basal and apical side, so they essentially have this columnar structure. You know, this is essentially how those look like. And then what you would typically do, you would typically image them in a confocal, but that's not really what you want to do these days. What you want to do these days is we can essentially image them with the arguably most advanced fluorescence microscopy technique that is available, which is called adaptive optics lattice light sheet microscopy. And so when you look at the evolution of microscopy, you can essentially think of it as like you have confocal microscopy, then you have Gaussian light sheets, then you have Bessel beam lattice light sheets, and then when you add adaptive optics to a lattice light sheet microscope, you essentially arrive at a system that can really go into a tissue, correct for the tissue-induced aberrations, and then get subcellular resolution at like deep in the tissue. And so we are operating at like 100 times faster than the confocal and 100 times less photo bleaching than the confocal. And the adaptive optics is really the game changer there. And when you then compare, like, you know, just confocal microscopy, you know, the reason it's called confocal is, you know, we essentially we produce this spot of light. It's essentially a pixel of light that we produce deep in our tissue. We created with the same objective as we are collecting the emission light from the pixel here. And so then if we want to, like, say, scan a three-dimensional volume, we have to essentially scan this pixel around in a line, then we have to scan the pixel around in a plane, and then we have to scan that plane in the 3D volume. So it takes on the order of like 100 seconds to like scan this whole volume. And then when you look at like your signal over how many volume stacks you can collect and how fast you're bleaching, this is essentially like how fast your signal exponentially decays with a confocal microscope, and our data rate is maybe about 1 gigabyte per hour. When you then move to lattice light sheet microscopy, you know, here we are essentially creating this very, very intricate light sheet pattern that is extremely thin, that we send with our excitation objective into our tissue. So that means we essentially illuminate one entire plane at a time. And then with our detection objective, we can detect this entire plane at a time. So that means if we want to like collect the data for a volume of tissue, we essentially just have to essentially like stack the plane of light through our tissue. So that gives us on the order of like 100 times faster image acquisition rate in our lattice light sheet. And then the fact that our light sheet is so thin, and the fact that we don't really have to essentially need these really high laser powers to create a confocal pixel somewhere deep in the tissue and then collect it back in the same light path, our photo bleaching rates are like, yeah, on the order of like 50 times lower than a standard confocal microscope. So it really allows you to collect like hundreds of these three-dimensional stacks before your sample bleaches, and which essentially gives you much more benign imaging condition. And then so the main game-changing ability then that we have is called adaptive optics. So typically when you image deeper into your tissue, you are collecting all these tissue-induced aberrations the deeper you go into your tissue. So what we do is we essentially borrow from astronomy. We essentially send a 2-photon guide star into the tissue, and from that 2-photon guide star we can learn exactly how the tissue aberrates our light. We can then you know, like, we take this light from our sample, we take a little bit from that light from our 2-photon guide star, put it into like into a Shack-Hartman wavefront sensor, into a control system, and then we can calculate the inverse of these aberrations and put them on a deformable mirror. And so the inverse of the aberrations on a deformable mirror plus the actual aberrations that come from our sample, they cancel each other. And then you get your corrected wavefronts directly onto your detector, which then allows you to really capture subcellular resolution deep in tissues, which really wasn't possible before. So then what that actually means is displayed on this slide. So on the left you see when we have switched the adaptive optics off. So what you see is essentially this haze and this blur of our tissue. And then on the right, what you see is these really nice Clathrin-mediated endocytosis spots. So what you expect is diffraction limited spots, so like 200 nanometer size diffraction limited spots, and that's really what you're getting with the adaptive optics. So the adaptive optics, can essentially punch through these tissue-induced aberrations, filters them out, and you can get the subcellular resolution back deep in the tissue, which is really unprecedented in microscopy in lattice light sheet. And so that's fantastic. And so that allows us to essentially collect data sets that look like this. So what you see here on the top left is our intestinal organoid. So we are imaging, so these are essentially these spherical structures, and we are imaging just the top of that spherical structure here. So this is essentially the image volume. We have in red the Clathrin, so the cage-forming molecule, and we have in green dynamin, which is the molecular scissors that cuts these vesicles off. And then so this is essentially our three-dimensional volume. And what you can kind of see is endocytosis happens on the plasma membrane, so on the outside of the cell. So that means when I'm spinning the data around, you kind of can see where the plasma membrane is just from essentially the endocytosis happening only on the outside. And so this is really like the end game data set for Clathrin-mediated endocytosis research. So typically you would have to do this in turf, and you would only have access to one plane. Here we have access to like the entire volume on the order of 30 cells simultaneously, where we can really follow the Clathrin-mediated endocytosis from the start of the inception of the endocytosis all the way from when these vesicles get endocytosed. And so one of the big problems then is, these data sets are fantastic, but these data sets are huge. So like standard microscopy, you like have essentially 2-dimensional plus time, 1 gigabyte per hour. There are essentially lots of scope software and tools to actually like visualize and quantitate and analyze these data sets. And for us, since we are creating 3 terabytes of data per hour, we essentially have to like create our own software and our own data analysis tools, since that data really doesn't exist yet. So part of my lab is really like computer science focused, where we create these data processing tools to quantify our data sets. And so that's one of the first tools that is available. So that's called PyLattice, for Python-based lattice light sheet image analysis. So you can detect these Clathrin-mediated endocytosis spots, and then you can track them over time, match the 2 fluorescence signals, and then look at lifetimes and these 3D movies. And so the way this looks like, so what I'm spinning around here is essentially one volumetric frame of our data set, where I have drawn a little square around each of the detections, essentially around each of the Clathrin-mediated endocytosis spots. And then, so this is raw data. What you want is essentially like you want to follow these individual Clathrin-mediated endocytosis spots. And what I'm showing here is our software essentially going into the data, segmenting these spots for each of these individual volumes, and then tracking them over time, and following those endocytosis spots over time. And so then when you do this and you essentially read the biological meaning, you can essentially do this in our entire image volume. So this is now in this gray. These are essentially the nuclei, just to give you like an idea of like us looking at these 9 different cells. This is a top view, this is a side view. I have color coded the length of these endocytosis spots that we can track. So some are like 250 seconds long, some are shorter, like typically they are like on the order of like 60 to 100 seconds. And then this is one event, and that a typical event, so this is essentially the fluorescence intensity over lifetime of this particular event. So you have Clathrin arriving at the site, building the cage, and then at some point Dynamon gets recruited when the cage is mature and cuts the cage off, and so the cage then is free to deliver the cargo in the inside of the cell to its destination. And this is essentially just statistics. You can do this for this entire field of view. Look at fluorescence intensity over time, and then you see this canonical Clathrin cage assembly, and then Dynamon cutting these necks off and then processing the cargo. And then you can obviously do this in multiple different conditions. You can do this for the basal, the lateral, and the apical membrane. You can see if there are differences between Clathrin endocytosis on the apical side, the basal side, the lateral side. If there are differences in lifetime, and then differences in different drug conditions, differences in when you have like genetic modifications. So this really allows you to do cell biology in all 4 dimensions. And so this was just like one toy, or like not a toy. It took us quite some time. This was essentially a validation example. We wanted to show that this technology is really possible. And so what I'm going to show now is a little glimpse into the future of what we are going to do next, and what we're up to now. So our goals are, we have to write high performance, extendable, user-friendly open source code to essentially enable the community. We're essentially building a new community of 4-dimensional cell biology. And seminars like this, where we across continents like talk to each other and build communities like this is fantastic, because we are really at the beginning of creating this new field. And so please join and let's build this community together, as we are just at the beginning of this field. And so the way I am moving this forward is so it's all Python based, and it's based in these Jupyter notebooks, so that also people with no computer science background can essentially use these libraries and load their data sets into Jupyter notebooks, and then play around with them, use the libraries to essentially like analyze the data. So this is essentially a segmentation library function that I've used to segment the data into the individual spots. So this is all on GitHub, so you can essentially download it, use it on your data, and please let me know if it works for you. And also please let me know if it doesn't work for you, because then we can fix it and make it better for the community. And so then we use machine learning because, if we are essentially use our human expert knowledge to segment the data, we can then essentially use that data as training data for machine learning algorithms to train the machines to essentially replace us and reduce the human bias that we have in data analysis. And so we typically use 3D convolutional neural networks because our data is inherently 3D. It's essentially like we have 3 space dimensions, 1 time dimension, and then we have channels like our essentially color as a 5th dimension. And so yeah, we use 3D convolutional neural networks. Typically we train on patches, and then like one big advantage here, if we train with neural networks, we can essentially lower the noise floor, or like we essentially can lower the signal to noise by which we can operate. So essentially, like classical algorithms, they need quite a high signal to noise ratio to actually like segment input data to these individual punctual structures. Okay, so but these deep learning tools, we can essentially train them very well, and they can operate even at like very high noise like levels. And so we can even recover these endocytosis spots in high noise levels. And so that essentially translates to, we can image the samples longer, and we can use laser powers that are even more benign to the sample. And so yeah, like we get essentially 5 times better retrieval with deep learning using those tools. And this is essentially just raw data. This is like the standard Laplacian of Gaussian filter. This is deep learning. So to convince ourselves that we're doing the right thing, and we can actually even recover data that we would have lost with the Laplacian of Gaussian filter here. And so yeah, we are essentially I'm writing everything so that it works on the Google Compute Cloud, so that non-computer science experts can just download our models and buy some time on the Google Compute Cloud and then run these models in the cloud without too much knowledge, essentially. And so yeah, it's a little bit, it's 5 times better than the classical algorithm. It's also 7 times faster, our machine learning solution. And so yeah, so what I really want to emphasize here is that we are at the beginning of this revolution of a new era of doing cell biology, because we are doing it in 4 dimensions live. And so yeah, our scopes produce 3 terabytes of data per hour. Our data pre-processing, like the deconvolution, de-skewing, we have solutions for that. But then for most of the biological information processing, so we are just at the beginning of creating the software. So for particle tracking, like proteins, drugs, we have solutions. And what I showed you is essentially the tools that exist. You know, PyLattice is one of the tools that exists. So that is essentially done. The next tools are going to be organelle tracking. So that's I'm now moving into organelles, and I'm moving into mitochondria. And so, but essentially there's also membrane tracking, nuclei tracking, and then entirely new biology that we have to do. And so we have to really build this new infrastructure for microscopy, and that's the new community that we're forming. And so I said that I'm going to move into organelles, and particularly I'm moving into mitochondria. And I have some more data about this, but yeah, we can essentially maybe talk about this in the one-on-one. Just this slide might be suffice. But so when you look at mitochondria, when you look at a textbook, this is not what mitochondria look like. Okay, so mitochondria actually are not these potato-shaped organelles. Mitochondria are also not what this 2-dimensional confocal picture suggests they look like. They're also not these just 2-dimensional filaments. In fact, mitochondria are these 3-dimensional filamentous networks that permeate the whole cell and are moving very, very quickly. Okay, so so this is essentially lattice light sheet data of mitochondria dynamics in stem cells. And so in the lab, what we're doing is we are tracking, we are essentially imaging and tracking the 3-dimensional mitochondrial network over time. And so what you can do with these new adaptive optics lattice light sheet microscopes, you can image tissues. In this case, this is not data from my lab, but this is essentially data that has been collected on the same microscope. You can take tissues and can image those tissues in 3 dimensions, collect these 3D movies from all the cells in your tissue, and then you can essentially move into your tissue, move into those individual cells, and then collect the 3D live dynamics of your organelles. And this was really not possible before. Like the adaptive optics lattice light sheet technology really allows you to do this, like 3D live in tissues. And that's I think what we are here for, all of us, to essentially investigate 3D tissues in novel ways. And so so this is the data that we can do. And so I'm just going to show like a couple more examples of what this technology can do, the adaptive optics lattice light sheet microscopy. This is not really data that is from my microscope, but it's data that has been collected on the same microscope, just to give you an idea of what's possible. So this is again Clathrin-mediated endocytosis in muscle tissue. So you can essentially, this is imaged a tissue, like you can computationally pull the tissue apart and look at all the individual cells in that tissue. And then you can take essentially a camera walk into your data set and look at these individual molecular events that are happening on the plasma membrane in individual cells in your tissue. And it's incredible, it's absolutely fantastic. And other data that you can collect, for example, would be something like this. Here, this is a human breast cancer cell in the blood vessel of a zebrafish. So in purple, you have a blood vessel, a capillary, and in green you have this human breast cancer cell, and you can image those breast cancer cells in 3D live, and you can see how they are crawling through the blood vessels and how they are probing the blood vessel walls for weak spots to essentially extravasate and form metastasis. And so this is like, you know, among the very, very first movies ever of the process of one of these cancer cells actually having found one of those weak spots and forming a metastasis right there. And, you know, I know that this data is going to, you know, inform how we think about cancer, how we think about all of these processes to really understand the fundamentals of what is happening. And yeah, and then just one last one, again, collected on the Janelia Adaptive Optics Lattice Light Sheet Microscope. And this is again, so in orange are all the plasma membranes of the zebrafish. And what we are looking at here is the outside of the zebrafish. This is the inside of the zebrafish. We are looking at, you know, this is one cell, another cell, another cell. And in the center, this is a lymphatic vessel. And there are, so what is in blue are essentially dextrane particles that have been, like sugar particles, that have been injected into the fish. And so what you see here, these are little immune cells that are essentially walking around, patrolling these lymphatic vessels and eating up these little sugar particles. And you see how dynamic everything is in a live animal and in live tissue. You know, like you see that, you know, obviously these live white cells, they are extremely dynamic, they are moving and are motile and are, you know, eating up these sugar particles. But at the same time, you know, like you see how the, like, quote unquote static tissue is obviously not static, but it's live, and the membrane is forming, reforming, and, you know, displaying all the membrane biophysics that we typically only see in textbooks, but now we have it live in 3D in front of our eyes. So it's incredibly exciting. It's incredibly exciting. And so yeah, so what I now have is essentially just, you know, we talked about me as a young professor starting at UCSD in the middle of the pandemic, and I essentially said that it was a blessing in disguise for us, because we started when we did not have the supply chain problems yet, so we could order all the parts, and many parts from Japan. You know, like our cameras are from Japan. All the high-grade optics parts, if they're not from Germany, they are from Japan. And so we ordered all the parts. The campus was very quiet because it was the pandemic. We had emergency authorization to, you know, build our microscope. And so this is just a time lapse of us building a microscope here. And so we built for the last, you know, couple of months essentially. And so what I have now is it's essentially done. We are extremely excited. So what I'm showing here is just a picture of the microscope, how it looks like. This is the camera that we used to collect the time lapse. What we are extremely excited about is displayed here on the screen. So the microscope is on at this moment, and it is displaying the lattice pattern of the lattice light sheet that we are essentially creating to image our samples. So the fact that we are essentially able to create our lattice light sheet in our microscope, yeah, we are extremely happy. It's working, it's online. And what I want to leave you with is, I think I've seen it. I think we are going to see 4-dimensional cell biology being the future. And we are moving from 2D cell culture, 2D imaging, and 2D manual data analysis to 3-dimensional tissues, to 3D live imaging of those tissues. So like 4-dimensional imaging that will create enormous amounts of data. And then we are going to use big data and machine learning tools to quantitate our data. And so we are very quickly moving from here to here. And I'm extremely excited to be at the forefront of building this community. I'm extremely excited to be here in this community to talk about this, and excited to form new collaborations. You know, I want to summarize that four-dimensional live imaging is a new window into biology. It relies heavily on math, computation, machine learning, and soon we will have it in organoids in our lab. And yeah, so I just want to thank everybody for having me. Like I'm really honored. Arigato gozaimasu for having me. And then this is just a little bit, yeah, we are looking for postdocs in our biology section. If you know somebody who would like to do mitochondrial biology in 4 dimensions on our adaptive optics Lattice light sheet microscope, please let them know and let them apply. Yeah, and I'm happy to answer any question. Thank you so much.
Thank you very much, Joe. If you have a question for Dr. Schoenenberg, please put it in the Q&A. While we're waiting, Joe, is there any possibility of doing lattice light sheet imaging with, for example, FRET labels or sort of markers of functional states of proteins? That what's the prospects of that?
Absolutely, absolutely. In fact, one of the reasons I'm so excited to be at UCSD, you know, we have the legacy of Roger Chan, of the first, you know, FRET reporters, and we have Jin Zhang here, who's building, you know, the latest generations of these FRET reporters in her lab, and like it works fantastically with these FRET labels. So yes, absolutely. Like we have 7 different laser lines. We can excite all these FRET labels. We have the filter sets. Please bring those. It is a fantastic application of the technology. Yes, yes, yes.
We might have to talk to you about that offline. We're running a little late, and I don't see any questions in the Q&A, but there will be a chance later. So at this stage, I'll thank you again and hand over to Kinoka Sensei to introduce the next speaker.
So thank you very much. I want to move the next presentation. The next presenter is Professor Katsumasa Fujita from the Department of the Applied Physics in Osaka University. And also he is director of the Photonics Center recently. And he graduated from the Department of the Applied Physics in 2000. After that, after getting the PhD, he becomes a research fellow, and he joined to the Kyoto Prefecture University of Medicine, and he come back to the Osaka University from the 2002 as the assistant professor. And 2020, last year, he became the professor. So today, he will show some optical microscopy and optical techniques to understand the biological phenomena. Okay, so the Fujita Sensei, please start.
Thank you very much, Professor Kinoka. I'm really happy to be here and share our research about Raman microscope, which we've been working on more than 10 years. So okay, I'll start my presentation. Oh, sorry. I think my slide is now showing.
Okay.
Okay, thank you. So so today I'd like to introduce our new development of microscope and also the application using that. And as interested, I'm from I'm in the Department of Applied Physics, but I did a postdoc in medical school. So I'm interested in both developing microscope and also do some application using that. And so I've been working on this research microscopy from when I was undergraduate student. And my interest is to build a microscope that did not exist before. So the microscope that can make invisible visible. So there are many choices of optical microscope as you can imagine. So I think this is one of most interesting part of optical microscopy study, since there are many different effects of light, interaction of light and the material, so different in different phenomena. So like as you seen in the previous talk, so fluorescence emits light from sample, and collecting this fluorescent light, you can show up many structures in the cells. But the image of microscopic image is very different in using different optical phenomena, like optical absorption, the phase, the scattering. So using different optical phenomena, you have different contrast. That means you can get a lot of different information from this optical microscope. And the recent advancement of optical microscope actually shows really a variety of information from different type of biological samples, from tissues, the cells, and so this is one of the introduced the previous speaker, lattice sheet, super resolution. So those are actually really important too in the biology now. So there are so many different types of microscope, but today I'd like to focus on this Rama Scattering microscope, which I've been working on. And so this is relatively a new way to image biological samples, and people are still investigating how to use it. So today I'd like to introduce what is this Raman scattering and the microscope technique, and want to discuss the future direction of this technique. So what is Raman scattering? So before starting seeing the microscopy part, I'd like to introduce the interaction of light and the molecule. And this view graph shows the energy state, so-called energy state of molecule. So their molecule can take several different energy level. The ground state, the lowest level, and there's a small increment from the ground state, which is called the vibrational excited state, which corresponds to the vibration of the molecule. So if the molecule vibrates, the energy goes up. Then the light, like the instant to the light, to the molecule. So the light and the molecule and overlap. And in many cases, they actually don't interact each other. So the molecule, light comes to the molecule and go to virtual state, which shows the overlap of light, photon, light, and the molecule. But light is just released, scattered, and the energy, there's no exchange of energy between them. So however, when Raman scattering occurs, after interaction, at the impact of light on the molecule, some part of energy remains, and the photon, the light leave from the molecule. So the energy moves to the molecule, which excites the molecule to this vibrational excited state, and which depends on what kind of structure the molecule has. So by observing this change of the energy, instant light and the scatter light, we can understand, we can see what kind of vibrational state is excited. And we can monitor this change of energy of light by using the wavelengths, because the energy of light is proportional, inversely proportional to the wavelengths, lambda. So by measuring this scattered light wavelengths, we can see what kind of molecule are existing in the sample. Okay, so this was discovered in Raman, and so it's known as Raman effect. And after his report in 1928, just 2 years later, he actually won the Nobel Prize. And this is an example of Raman spectrum we obtained from some cell sample. And we shine 532 nanometer excitation wavelengths here. And then we detected from 540 to 640 nanometer. And you can see many different wavelengths of scatter light. And this is a mixture of Raman scattering, also fluorescence, for example, but those sharp peak shows Raman scattering, which can be assigned to different molecular structures. Then in Raman microscope, we detect this Raman spectra, and if we define each part of the sample. So this is the cell sample, and by measuring Raman spectra, we have this 3D data set showing the Raman spectra, and which cause this the spectrum direction Raman shift, because the energy is changed and the wavelength is shifted. So this is the spectral direction, and this X, Y is the spatial directions. And this, after obtaining this 3D data set, we pick up the molecular vibrations that shows wave shift, or wave number shift, like here. And so these are examples. The 750 wave number, this can be assigned to cytochrome spiral ring breathing mode. Especially this is seen in HeLa cell strongly in the mitochondria, because cytochrome C is highly concentrated in mitochondria. And the 1683 is a vibrational mode peptide bond, and this is called amide 1 bond, and especially richly seen in protein beta sheet. So the protein exists everywhere, and the signal exists everywhere. The 2850 is CH2 stretching mode, and the CH2 is included in lipid molecule richly. So we have the contrast of the lipid droplet and the membrane of mitochondria. So we can obtain those molecular contrast of the sample without labeling, so just shining light, and they create the image that shows the distribution of this vibrational modes. The Rama scattering was discovered a long time ago, but it has not been used in biological research because this effect is very, very weak. And this is a comparison of the cross-section, which shows the efficiency of light-material interaction in case of Rama scattering and light absorption to the fluorescent molecule. So as you see here, this efficiency is so small in Raman scattering. That means you have to wait a long time to correct the signal light from sample, or you need a high power laser. But the recent advancement of high sensitivity detector and laser technology on optical components, now we can use this in Raman in biological applications. And in addition, we actually try to increase this speed. Since to obtain this kind of scattering, we have to shine the laser single point, and then we have to detect the spectrum from the spectrophotometer. And so in the moderate laser power, we need a few or several seconds for spectrum. And if we want to take 120 to 120 pixels, it takes about more than 20 hours. And so it takes hours to get one image. So this is too slow for living cell imaging. So we in our development, we use the line illumination, which to shine the sample in one line-shaped focus, and Raman spectra from each position of line, emitted line, can be detected at the same time. So this simple improvement actually can improve imaging speed drastically. So in the same under same condition, so the 23 hours become 11 minutes. So we can. So this is still slow compared to fluorescence imaging. However, we can perform a lot of experiments and to be used in biological researches. And this is an example, another example of Raman image of living HeLa cell, and again cytochrome. And this is the other part is phenylalanine, showing the phenyl ring angular bending. And the phenylalanine is also seen in commonly in the protein. You have this is a protein structure, this protein distribution in the cell, in the lipid here. And those images are taken just 20 minutes. So, so this is still slow, however, we can now use this in some biological studies. And after development, so we actually start the application. So this is actually the opposite way, because since I'm from physics department, so I start from this technology development, and then think how we can use this. But the Raman spectroscopy itself is very well known and powerful technique in the analytical chemistry. So we can combine analytical chemistry approach in the cell biology. Then we are now doing those 3 different applications. And I will start from two of them first. And since we detect Rama scattering directly from this molecule, so we don't have to stay in the sample. So this is totally library-free imaging. And from the spectrum, so we can analyze the cell. So those two are a new approach in biological imaging. And especially we found that the spectra shows strong peak of cytochrome. And in a HeLa cell case, this is very strong in cytochrome C in mitochondria. So we performed the application of the observation of cell signaling during apoptosis, which is a release of cytochrome from mitochondria after activation of apoptosis, and it's called induced caspase cascade, and it makes this kind of destruction of cells. And so we successfully imaged this signaling process of cytochrome C mitochondrial release, and the cytochrome C release from mitochondria, and that is here. So the change of that spatial distribution of cytochrome C signal is clearly seen. And we also applied this to monitor the dysfunction of mitochondria using RGC5, which is the retinal neural cell line. And it is known that under glutamate stress, this cell line, neural cell is damaged. So usually this is measured by using fluorescence or some destructive way, but by detecting the reduced cytochrome C signal, so we can see the decrease of the signal because of damage. So this is due to the oxidation of cytochrome, and which is the Raman is also sensitive to this redox state. And we compared this decrease of Raman peak, conventional MMTT assay, ATT assay, ROS assay. So those are corresponding well. So we can perform those assays without library and even in a subcellular level. And so this is the library imaging of the cytochromes. And we're also interested in using this whole spectra because this spectra shows many different cells, molecules, and it provides the chemical balance of the cell, like how much protein, how much lipid, nucleus. We cannot tell exactly, but so the balance changes the spectrum shape. So we are trying to use this spectrum shape to characterize cell species and conditions. And so this research is a collaboration research with Riken in Japan. And those Taro, Hide, Tomo, those 3 people are interested in separating the cell states of the ES cells. So after observation of Raman, we can see some difference in image, but it's not really quantitative. So to compare them quantitatively, we compared the spectrum and they applied principal component analysis, that is mathematically separate the spectral component, and they compare the spectrum shape of each state of the cell differentiations. And this is J0 and to the day 14. And as you see here, so this the 2D map shows that spectrum variation of the cells. So each point shows the cell. So in this J0, each cell separated in space. That means they have different spectrum. However, after the differentiation start, they start to go to the same positions. That means the cell chemical component in the cell becomes very similar. So this differentiation, during differentiation, we can tell how much the chemical component changes, chemical balance changes, and which can be used to evaluate the cell differentiation. So this research, kind of research, has also done is the mitogenetic differentiation and also T-cell activations, and that we are even tried more applying to different type of cells. And I think I'd like to introduce another application, small molecule imaging is Raman. And the fluorescence microscope is very useful, but they actually cannot be used to some molecules. Like those small molecules, actually too small to be labeled by fluorescence molecule. So add this fluorescent molecule into a small molecule, the chemical property of them changes. So usually for small molecules, fluorescence imaging is not used. So however, the Raman can detect them, but as you see here, there are so many different kinds of molecules in the cell. We cannot really separate them clearly. So some strong signal can be separated, but we cannot separate all the molecules. So we try to use this region, which actually is very silent. That means those not many Raman peak appears. So we used this region by using the alkyne. This alkyne is a triple bond between carbons, and this carbons, like alkyne, shows the Raman peak on this region. And we propose the use of this alkyne as a tag of small molecule. So since this is small, so we can use this as a tag of molecule, which can be used to specifically image by Raman microscope. And this is Raman spectrum EDU, which has it's alkyne in EDU, and you can see strong alkyne peak in this cell silent region. So we can detect by using this alkyne whether this EDU exists in the cell. And as a POC, we introduced alkyne with origin EDU into the cells, and the cultured cells with EDU. And since EDU is has similar structure thymine, we can expect this into localized in the nucleus. So this is the experimental result. Raman image of hair cells, which has been incubated with EDU, showed a strong signal and alkyne in the nucleus, while no treatment with EDU, there's no signal. So this demonstration shows that the alkyne signal can be used to detect EDU. And the advantage of this alkyne is the size. Of course, it's much smaller than the other staining methods. And we also performed Raman tau imaging for the observation lipid domains, the drug, and also organelles. And since this is new modality, now we are trying to exploring what kind of molecule we can observe in the cell. So this is another example. The EDU and the ubiquinol, both are using a different type of alkyne, and then they image simultaneously. So like fluorescence multicolor imaging, we can also perform multicolor Raman tau imaging too. And these activities there are many groups are starting to using that. And since the small molecule has not been able to be imaged by conventional Raman fluorescence microscope, so now the community are trying to investigate so how this is useful in cell cell biology or medical sciences. Okay, so I think I'm running out of time. So I just finished by saying one issue. Since the Raman tau is useful for imaging of small molecule, however, still there's an issue because of the weak Rama scattering efficiency. We need a high concentration molecule inside. So in this, the experimental result shows we need about 100 to 200 micromolar of molecule in the cell. So this is still high. This is actually high concentration for the molecules in the cell. So we are now trying to improve the sensitivity of this detecting Raman tau, and hopefully we can show more results showing different type of molecule in the cells. Okay. So yes, so I'd like to finish my talk. Okay, so this is collaborators. Since I'm just building microscope, so that those applications has performed with many collaborators. And thank you very much for your attention.
Thank you very much, Professor Fujita. So the audience, if you have some question, please put that Q&A. So just I have a comment first. Thank you very much, Mr. Sensei, and you prepared the many variety of the application using the Rama microscopy. So the, how to say, in your case, how to say, still time lapse is not sufficient, as you mentioned. So what is the next step for the, how to say, time lapse? Do you have any idea to get the, how to say, more dynamic behavior?
Thank you very much. Yes, I'm trying to increase the sensitivity, and if the sensitivity increase, we can shorten the imaging time. The one of the way is using some enhancement effect, and the so-called surface enhancement effect, which is seen on the surface of metal. So we're also working on this.
Sensei, do you have any question for him? We want to move to the, how to say, overall topics.
I think both of these speakers have really shown us technologies that are very transformative, and I'm sure there's going to be a lot of interest in collaborating with both of them. And we also have Professor Ling Yan Shi here at UC San Diego working on stimulated Raman spectroscopy, and the potential to be able to image live cells and tissues, get their detailed dynamic structures and their molecular and metabolic activity really is transformative for cell biology. And in addition to the sort of large-scale data analysis and AI tools, coming from my perspective, what these data will be particularly useful for is developing and validating computational models. So if either of the speakers has any comments on, the use of these data with computational models, or to develop a new class of much more detailed computational model of cell biology, I welcome your comments.
Okay, yeah, so let me start. So as you see, as I introduced, the Raman spectrum has a lot of rich information on molecule. Also, resolution is sub-micron scale, so that the data set is actually huge, and we have not explored what kind of information really included there. So I think both approach, like a supervised and unsupervised analysis, actually we can tell more about biological phenomena. I mean, we just like I always think imaging. Many people is using imaging to confirm what people believe exist. Yeah, but taking this kind of Raman spectroscopy way, we can find something new in the data. So that combination of this unsupervised and supervised method will tell you more about the biology or the mechanics of the cells or tissues.
Thank you.
So I think from my perspective, we're actually using simulations a lot to create essentially synthetic data sets for our machine learning tools. And I think there are huge opportunities for simulation because it, like, the 3D live imaging essentially creates experimental data against which simulations can be matched. But then at the same time, we can also, create simulations that cross modalities. We can essentially take the fluorescence movies and then create simulations that say use mitochondria 3D networks essentially as a morphology blueprint, and then essentially simulate the more molecular dynamics or the more coarse-grained protein dynamics in those three-dimensional morphologies, to then essentially simulate and advance the molecular scale a little bit, and then go one more step with the larger scale fluorescence dynamics, and then essentially similar to like how molecular dynamic simulations and QMM simulations do their bridging between scales. Essentially like Raman spectroscopy can essentially go much deeper into like molecular size than say fluorescence microscopy, but we can essentially maybe be faster. And so I think simulations has a fantastic role there to bridge and to unify.
Well, I'd like to just thank both of our speakers again for really outstanding and exciting talks that I'm sure will spur a lot of discussion and add to the depth of collaborations between UC San Diego and Osaka. Also want to use this opportunity to thank again Kimura-san, Waga-san, Hyuchi-san, and the LinkJ staff for organizing and hosting this webinar series. And if there are no more questions or comments from Kinoka Sensei.
At this time, no, but I hope if possible, please join to have a good collaboration, especially for the Fujita Sensei's, I would say, understanding the phenomena based on the chemical reaction, and also the Joe having some tracking system to predict the future, further unknown phenomena. I think so. If you have some interest in geozer, it's much better to create a new field, I think. Thank you.
Over to you, Miwako.
Thank you very much for wonderful moderation, Kinoka Sensei and Andrew, and also wonderful talk, Fujita Sensei and Schoenberg Sensei. And it is very enlightening. And I was very impressed by both speakers. And, you can see, you can believe what you see. And it's amazing, it sounds like, is this a movie or the real world? And I cannot believe how advanced the technology is. And this will really help other scientists to better understand the biology. And as Joe mentioned, this, building community. And he was very impressive that he said that this is the beginning of a new era. And that is an exciting thing. And we have been, been working in the dark for the last 20 months or so. And I kind of feel the light for 2022. And I do hope that we'll be able to meet in person and talk. And also, of course, seeing the equipment also, I think very educational as well. So I do, my best in my role to facilitate the collaboration so that, we'll achieve wonderful research and also educate the next generation of students and researchers and scientists. And with that, I'd like to have Kyuchi-san to join us and for his closing remarks. He is not here. Okay, he's coming. Okay.
I'm here. ご登壇いただいた先生方、ご視聴いただいた皆様、誠にありがとうございました。今後もリンク J と UC サンディエゴはジョイントウェビナーを開催してまいります。お聞きになりたいテーマや登壇者についてご意見、ご要望いただきたく、ウェビナー終了後に表示されるアンケートへの回答にご協力ください。アンケートは15秒程度でお答えいただけます。リンク J からイベントお知らせメールを受け取りたい方は、表示されています QR コードからご登録ください。その他お問い合わせなどはリンク J ホームページからご連絡ください。本日はありがとうございました。
Thank you. Bye-bye. Thank you. See you next time.